I thought we could talk about high dimensions and data some more since we talked about it a lot. I feel no need to get through any of these slides. So just interrupt me. I'm going to throw some hand grenades, hopefully. So let's start with the first one. This is my favorite quote. This is by a biologist. And I kind of just like felt this throughout the day yesterday, which is this, this idea that there is like a fundamental aesthetic disagreement emerging between the physicists and everyone else about whether what's limiting us is more data or what's limiting us is maybe better frameworks for actually working with the existing data. And I'm squarely in the second camp. It's very hard for me to believe that taking more data will really help us answer the questions we want. But you got to put up and shut up. So here is a series of things. This is not to tell you anything. Mostly pay attention to the pictures. So this was done by my student Maria. It started a long time ago. We started thinking about this stuff in 2012, back when things happened. We wrote some models and it was right when single cell RNA-seq came out and everyone's like, you can't use any of this data. It's all useless. Though as far as I can tell, it was still perfectly good. And the stuff I'm going to tell you about today, we can't get published, but I still think it's the best thing to come out of the group in the last 6 or 7 years. So I'll start with, this picture. Usually I got beat to it, but usually what I like about this picture is that there's two of them. And I think actually the picture this picture is more informative than the other one, because the whole idea is, again, as was stated earlier, it's the emergence gambit. But as was also pointed out earlier in the talk, there's something very annoying about this picture. And it's mostly that I look at this, and I'm a physicist or a statistician or any quantitative person. The first thing I tell my students is you never produce a graph without some scales and some axes. And it is extremely confusing what the scales and axes are. Right? From Jin Wang, we saw that maybe we could think of this as a parameter that changes an interaction coefficient, but it's extremely hard, right? And that's because we don't understand this. Maybe we don't understand this high dimensional geometry, but it's not just Waddington. At least Waddington was conceptually and philosophically interesting. I would say I have the same question about these data sets, which is that this is some data that I'm going to show you later. This is a data set that's been shown multiple times already in this meeting. This is a Lone Klein's barcoding data set on homeopoietic stem cells. This is big. I was forced onto lung development because I found some experimental collaborators I really, really love. They're doctors. They want to cure children, but somehow they care about science because they want to cure children. So it's been really fun working with them, and it's on lung development. So I'm going to show you a lot of data from a lung development data set, atlas. And all you have to understand is mostly I'm going to focus on these kind of cell types that are genomically very similar, but not very similar. Most of the data I'm going to show you, just because it's a 20-minute talk, is going to be about progenitor cells turning into two closely related cell types called AT1 and AT2. They're epithelial cell types. It's not really important, but we're going to look at epithelial lung development. What's important for all this data is that atlases now more and more have embryonic time points. So you don't just get a snapshot, you get time, developmental time, right? Not pseudo time. And so what I'd like to know is how do I visualize all this data where the coordinates have some meaning and the heights have some meaning, and take the high dimensionality seriously. I don't want to filter any genes. I don't want to look at variability. I don't want to do anything. All right. The only thing I'm going to talk about is normalizing. So the whole Python code that makes every plot I showed you is maybe 7 lines long, literally. And so this is what I'm going to do. So the question is, need to think about how to properly mathematically represent this and plot high dimensional data. And what that means to me is, how do we make meaningful coordinates? It is very annoying being up here. It is very hard. It's just, I like to point, but very confusing. Where to go? How do we make meaningful coordinates and scales? And so here's my gambit. Here's what I think. That development. Okay, I can't proofread my slides apparently when I make them while other people are talking in the middle. But the basic idea I want to say is that if I look at what biologically meaningful gene expression profiles are, that geometrically, we always think of them as a very complicated manifold, but actually, I think it's very reasonable and works very well to just treat them as approximately linear. And I will show you the consequences of doing this. That it's not actually very complicated. We always imagine this very funny thing. And that the actual developmental dynamics is actually kind of complicated dynamics in a very simple geometry. And if I have time, I'll show you how we've been trying to combine. Yeah, go ahead.
I mean, isn't everything approximately linear if you look locally enough?
Okay, yeah, I agree. So if I look at all cell types in a mouse cell atlas, is that local or global? You tell me. You have to tell me the metric.
I don't think that's a manifold to begin with. So.
I agree, it's not a manifold. Subspace. It's not even a variety. I agree. I am using manifold in the machine learning sense where it's everything except for a manifold, as was said in the first talk. It's a variety, it's a graph. The one thing it's not is a manifold. I agree with that statement. Okay, I mean subspace spanned by gene expression.
I just think like maybe maybe this is true because you're thinking in a kind of local way and
okay, you tell me if it's local. It's going to get even dumber than this.
Okay.
What I'm going to do is even dumber than this. So you're going to get more unhappy. Right? But I'm going to make you plots. I'm going to plot data. All right? So here's the basic idea. All right? Basic idea is let's do linear algebra in a non-orthogonal basis. That's the whole idea. The whole idea is I have genes. They live in some high expression space. I have some directions that are special. They're special because some biologist called them a cell type in an atlas, or they're special because my favorite biological friend told me this is a special direction. I care about it. For the purposes of this talk, they're going to be cell type A, cell type B, whatever. And then what happens is I give you some number of directions that's less than the number of genes I measure. So I'm happy because now measuring 20,000 genes is a plus. It's not a negative because I can always find a subspace that's planned by the directions I care about. And then I basically just take my random sample and I do what's shown here. I project onto that plane, and then that projection gives me a coordinate system in these special directions I care about. So all the assumptions, as it was stated, assumptions should be made very clear. The assumptions are the special directions I care about. All right, so that's it. That's the gambit. So the gambit is let's plot atlas data this way. All right. There is one more thing that's important. We're not doing any harmonization. We're not doing any filtering. The one thing we're going to do is we're going to normalize properly. So what we're going to do is that I have never been convinced that single-cell RNA-seq has any information beyond rank information. So what I'm going to do is I'm going to normalize using something that I found out later is called quantile normalization, but I didn't know that. We didn't know that at the time because we're physicists and it seems like a strange name to call something to us. And what we're going to do is we're going to just take each cell. We're going to rank order the genes. So each cell is processed separately. Then we're going to normalize, and then we're going to convert it to a z-score, right? So if something's at 99th percentile, 98.6 percentile, it gets z equals 2. If something is at 30th percentile of expression, it gets whatever, z-score minus 2. Something at 50th percentile gets z-score 0. We take everything that's 0 and we just put it to the lowest z-score. It's not super sensitive to that, but I am all at the lowest z-score, right? What that does is it takes every vector, because it's normally distributed, I'm going to exploit high dimensions again, because if I take a random vector drawn from a Gaussian and I take another Gaussian random vector in high dimensions, they're orthogonal to each other. So what turned out to be a curse actually turns out to be a blessing, because basically now if I see any projection on something else, I know it's probably not by chance, right? All right. So the last thing is, how do we construct these special directions I care about? And there, single-cell RNA-seq is very, very noisy, right? So I want to construct these vectors that correspond to directions what I care about. So all I do is I pseudo-bulk count and I normalize the same way. Yeah, good.
Stupid question. So the previous slide, you said everything linear. People you know this better than me, like in the machine learning thing, when they try to figure out how to read what language models are doing, they sort of try to do this thing where you have non-orthonormal bases and you try to represent information. But the way the things work there is you have one non-linearity. You have a ReLU function, for example, and that helps you take more complex data and put it on a non-orthonormal.
I'm not doing any of that.
Okay.
I'm doing linear algebra with these two normalization steps, throwing out I'm even going across platforms and labs, and I'm doing no harmonization, nothing. All I'm doing is quantum normalization.
I was
That's my
wondering,
harmonizing.
one of these things would start to look like a ReLU.
I don't know why they would look like a ReLU.
I mean, possibly a little bit just because of the dropout, honestly. It might be a bit
I mean, they're
like,
put at -2. I mean, you can pseudo bulk it, it all works. So it works with pseudo bulk better than it ever works with single cells, right? But the point is I can do this with single cells.
Do you filter anything from the pseudobulk? Like you sometimes in pseudobulk you
100 cells.
use sum over no in terms of counts, like you sum over noise, and because you have low counts across many cells, and then you end up having like a bunch of
Yep. As long as we normalize after we pseudo bulk, after we sum the counts, instead of normalizing then summing the counts. So we sum the counts literally, and then we just quantum normalize. That's what we do.
and consider that you're gonna sum some noise in that. No,
I surely am.
just
I surely am. I surely am going to sum some noise, but the hope is that the directions are.
cells.
I mean, the point is that what happens is you can figure it out. The only thing that pops up when I take a non-orthogonal basis is the correlation between cell types, like these vectors, right? So what happens is it's like a normal thing, except I have to invert one matrix. The rank of that matrix is set by the correlation of cell type A, B, C, D. As long as they're not so degenerate that that's 0, everything seems to be very robust. So there's only one matrix inverse that sets everything. So the basic claim is that this basic geometry can capture lots of stuff. And we claim, we spent a lot of time doing this. We spent a lot of time for various reasons. I got irritated about this transcript former model a lot. So we spent a lot of time. My student Juan told me to stop complaining and we'll just do something. So you can look for yourself. We put a preprint, will never get published. I'm pretty sure. It's now in PLOS 1. I'm sure it's going to get rejected from there too. Where we use zero parameters, and we can beat these foundation models, including the ones that were shown earlier, on almost all downstream tasks. Not always, because we don't fine tune and we have no parameters, but the more out of sample it is, the better we do. Let's put it that way. The more I have to generalize to new settings, the better linear stuff works. And it might be because this data is extremely noisy, or it might reflect something about the biology that I can't tell you.
But can you just say what is the task on which you're outperforming the
cell type classification? I don't know. Go through. They have a set of tasks like inferring a gene network.
what does it mean? Sorry, I think it's a little confusing just because you say it's parameter-free representation. That's fine.
But I just do linear algebra. It's cell type classification. Let's do that. So what I do is I take a random gene expression profile. I can now calculate its projection on different cell types, and I assign it to the cell type that has the highest projection. It's linear algebra. Right? But the interesting thing is I can make these plots. The only thing I find interesting about this is let's plot data. Right? So, so I can either start with data like this. Right? This is some data. It's a diffusion map, it's a U map, or whatever. But now all I'm going to do is I'm going to take my axes. So often we take our axes from one experiment or one thing and we plot them from another one. So here are these 2 points, AT1 and AT2. These are, some ground truth labels made from somewhere. And what I want to point out to you is the axes are going to change, AT1 and AT2. If this was pseudo bulk data, you'd get projections around 0.9 or 1. 1 is a meaningful number. That's like a perfect projection. And what you can see is like, okay, the AT, what's going to happen is I can look at different things. So I'm projecting different cell types. These are all epithelial, terminally differentiated stuff. And I see that AT2 cells lit there and AT1 cells there, but I can look at a different 2-dimensional plane through this thing. So if I now plot ciliated against AT1, you see the AT1 stays here, but the ciliated pop up. With meaningful axes, meaningful distance metrics. And what's fun is let's watch lung development. All right, so here is a very dumb thing. It's just the lung cell atlas. They just sequence cells at different time points. So embryonic, E5, 16, 18, P0 is post-birth. And let's watch a movie. So here's a movie. It's just a 2-dimensional projection. I can give you scores for everything else. And so you can see I start at 12.5. I don't project on anything. All the cells, every data point is a cell. And you get this fun thing where it seems like the cloud moves in this intermediate AT1, AT2 things right around post-birth. You get this kind of, intermediate population that sits here and then kind of disappears. All right. So we can watch that in 2 dimensions. We can watch that in 3 dimensions. Maybe you'll play. Maybe. All right. Oh, there it goes. Right. So here's a different direction that's called early epithelium. It's just a progenitor cell. So you see it projects on that, kind of goes down here, seems to go to an intermediate stable and bifurcate. Right. So just plotting data using linear algebra. No fits. No normalization. No, nothing. Here is what it looks like. And you know what looks fun to us is that this looks like an intermediate. If I look at this red point, it looks like an intermediate saddle to us. It looks like something where I'm going towards something. It gets some population. It gets stuck there for a little while and then kind of disappears.
So when you, good. Yeah, how did you get these bases and how did you name them?
I named them because someone in an atlas told me this was called AT1. I really got them because my collaborator Daryl and Laertes said, this is a good data set for AT1. This is a good data set for AT2. Pseudo bulk those. That's all I did.
Oh, so you had the cell type labels, you pseudo bulk.
Yeah. I mean, every cell atlas comes with cell type labels, right?
I see.
Every experiment comes with labels. I'm not going to pretend. Yeah.
So I had the same question. Like,
yeah,
you're not choosing a basis. It's just taking what people told you and plotting along this.
Yeah, I mean the base. I'm not choosing the basis. The biologists who are much smarter than me are choosing the basis. Why should I cluster to choose a basis when I have 60 years of biological knowledge to tell me what the important directions are? Yeah, I got it.
Well, because de novo cell type discovery is important, right? Like, so there are many cases where you don't have a cell atlas and you don't have a reference, right? So
okay, but you have a hypothesis for what a cell type is, and then what we do think about this a lot, actually. I'm being facetious. We spend a lot of time thinking about basis, but one of the things you can ask is, if I add another basis point, we could talk about this, but like, for example, if we want to know whether we're in a deep hole about whether something is a diseased state is another basis type or not. And once you have time series data, you can actually see by adding an extra axis on the projection and seeing how things change with time, you can come up with, you can do the same thing. You can basically say, let's pretend this is a cell type that I don't know. How robust is, for example, the developmental dynamics, or how robust is this across data sets? Is it change the position in all the other axes or not? And by using metrics like that, you can actually ask these questions. But it's like, it's the same thing. Like at some point you cluster and decide or you do something to decide. But that's a weird space. You want to use a different space to evaluate whether you think this is real or not just cluster in UMAP and then say it's close to the UMAP. And so it's another cell type. Yeah. If you give me 1 minute, I just want to say one thing, right? And then I'll be done, which is that I think the key idea of this is that if this geometry is simple, then what I can do is I can use this simple linear geometry and then define models on it. And so what's fun is that we could take this same basic geometry, gene expression, cell fate, and then you give me a bifurcation a la Eric, who Eric will tell you. And what's fun is that we can basically write a model, which is not quite Eric's model, but a generalization. And this is what our models would do. And these are what real life data looks like. And so you take a bifurcation class that Eric will tell you about. The top shows you just model predictions of what this will look like. And this is what real data looks like. And it seems like, I'm not going to say we can identify them, but it looks like there's signatures of these kind of bifurcation classes that Eric is talking about in the time developmental dynamics of these things. Okay, I'm done.
Question there, and then we'll.
Yeah. 1906. I've been timing.
I know.
Picking up on Vijay's point, so cell states, cell functions, cell archetypes are kinds of all different things that you could also think about using to specify a basis here.
Yep.
How does your idea connect to archetypal analysis, finding kinds of the points, using those as bases, and within individual cell types, projecting onto those, trying to.
Oh, I don't think it's too different. I'm just saying do linear algebra. And what's fun is that here I've focused on cell type, but now we're starting to play with things like stress. Is there anything that you think corresponds to a pretty well-defined direction? Right? Where this starts breaking down is when things are not defined by a direction gene space, but with a cloud that can't be defined by a series of vectors, right? So if I have a lot of structure, I can't think of it as an ellipsoid. As long as I can think about it as an ellipsoid, I can write it as a series of vectors that I can put it in. But once you condition enough, almost everything looks like an ellipsoid. And based on how noisy the data is, it doesn't seem to matter. But yeah, I agree. There's like subtle, all the questions have been pushed into how I choose this. But I view that as a feature, not a bug, because the assumptions are extremely clear.
Okay, so just in the interest of time and because we got this request from the staff, we will move on. Let's all thank Pankaj for the great talk.